Hello everyone, welcome to my channel. Starting from today, I'll be doing the 7th lesson in ALL biology section, molecular biology and recombinant DNA technology. So, when you get into this lesson, as a whole is discussed about DNA. Okay, how DNA is synthesized, how replicated, and different applications of DNA as we desire, even PCR. You must be very interested in knowing about PCR because it's very important these days, right? Anyways, first of all, we have to know why DNA. Almost every living organism possesses DNA as their genetic material. Actually, only few viruses will have RNA as genetic material. All other organisms have DNA as their genetic material. So, there must be something important in DNA, right? So, in your resource book, we have 3 definite reasons. So, you will have to remember them. So, if you have the resource book, please highlight or underline that. Okay, first reason is the DNA replication process is very accurate. Accurate means very correct. It's like 100% error free. In the next video, we will discuss about DNA replication. So until that, remember that process is very accurate. And the second reason is genetic information can be transmitted from generation to generation. That means DNA can transmit genetic information from generation to generation. And the third reason is DNA can store and express genetic information. So to clarify the second and third importances, I will give you an example. Think about the color of your eyes. That means color of the iris. So I am having black eyes because my parents have black eyes, right? Most of Sri Lankans have black eyes. That means the gene responsible for giving the black color for iris is transmitted from my parent generation to my generation, right? So that's why I am having black eyes. But just having the gene won't express the phenotype. For giving the phenotype, genes must be expressed. For that, we need protein synthesis. And even in protein synthesis, we need DNA. Okay, we will learn that in the future too, the process of protein synthesis. So that's how DNA is that much important. Okay, next, we will have to go through your memory in the second lesson. Okay, second lesson you already learned a lot about DNA's molecular structure. So let's go through a recap very soon. So now I'm going to discuss the Watson and Crick model. According to them, DNA has a double helical structure. You see, there are 2 strands which are arranged in a spiral way. So that's why we call it a double helical structure. And also notice that one DNA strand I've named from 3' to 5' and the other one from 5' to 3'. Due to this nature, we call the 2 complementary strands in the DNA molecule are antiparallel. They are parallel to each other, but one is the upside down of other. So we call it antiparallelism. Also, you can notice that they've given the names that this looks like a spiral staircase and a twisted ladder. If we think that this is a spiral staircase, you must have the rails and the steps. So the rails are the backbone, which is made by alternatively present phosphate groups and pentose sugars. And the steps are the nitrogenous bases. To understand it even more clear, let's look at the molecular structure. So in this molecular structure, you know that deoxyribonucleotide has 3 main parts: the deoxyribose pentose sugar, the phosphate group and the nitrogenous base. And even you know there are 4 types of nitrogenous bases that can be present in DNA: adenine, thymine, cytosine, and guanine. Out of them, you know that adenine and guanine are purines with 2 carbon rings. So I have represented it from a little bit lengthier shape. And pyrimidines have just only 1 carbon ring. So cytosine and thymine are pyrimidines. Okay, now see that. Let's look at that 5' and 3' thing. You will understand that if you see how the carbon skeleton is numbered. So see 1, 2, 3, 4, 5. So see, this strand ends from 5th carbon. So we call it 5'. And here it ends from the 3rd carbon. Here it's the 3rd carbon. This one is the 3rd carbon. So we call it 3'. And as this is antiparallel, so it starts with 3' but ends with the 5th carbon. So we call it 5'. So that's it. And also you know in complementary base pairing, purine always combine with the pyrimidine. And even adenine and thymine are bound via 2 hydrogen bonds, while cytosine and guanine are bound via 3 hydrogen bonds. So that's all for the molecular structure of DNA. Also remember that T.H. Morgan and his group discovered that chromatin are made up of genes and genes contain DNA. Okay, now let's learn what is called architecture of chromosomes. You know, in eukaryotes we have a nucleus, but in prokaryotes they don't have a definite nucleus. So the area where genetic information is present in prokaryotes is called the nucleoid area. So how DNA molecules are arranged in this nucleus or nucleoid area is called the architecture of chromosomes. Okay, now this word chromosomes, for both prokaryotes and eukaryotes we use the term chromosomes for packed DNA, but only true chromosomes are present in eukaryotes. So you have to remember that. But we generally call them chromosomes. In both prokaryotes and eukaryotes, the DNA molecule is a single DNA. One chromosome has a single DNA molecule which is a double helix. The difference is the way they are arranged. I mean, in prokaryote cells, we have circular DNA. The double helix, the single double helix is arranged in a circular manner. But in eukaryotes, they are arranged in a linear manner. So that's the difference. And there may be certain other differences, you know, in eukaryotes, the genetic information storage must be much complex, right? Do you know that DNA are so long? Even there may be DNA of length like 100 meters and thousands of meters, okay? Though they are very tiny, they are so long. So, how can you store that much long things in very tiny cell? A tiny cell which you can't even see in the naked eye. So there must be a very good mechanism to package them, right? So we will have to learn about DNA packaging. Simply, DNA packaging is the containment of genome. Genome means DNA, okay? Containment of genome within the nucleus if a eukaryote or the nucleoid area of a prokaryote. So in general we can say DNA packaging is the containment of genome in the nucleus or nucleoid area of an organism. So that's called DNA packaging. So we will have to find out how the DNA packaging happened in prokaryotes and eukaryotes separately. So let's go through the prokaryotes first. This diagrammatic representation will help you to understand DNA packaging in prokaryotes. So here I've represented just a circular unfolded chromosome. That means the double helix. The single double helix is arranged in circular manner. So see, it's like 350 microns in width. But in the second step, this is coiled with association with the RNA and protein core. So protein helps DNA packaging in prokaryotes. So see, here this is coiled into loops, and the size is very much reduced, like more than 10 times, right? So here size is reduced into like 15 times less. And how has that happened? These coiled loops has been supercoiled. See, these are supercoiled into domains. The small unit is called a domain. Okay, and this RNA and protein core is very important in this process. It attaches the chromosome to membrane. Also, it acts as a barrier for rotation. These loops cannot rotate as they want because they are bound to the central core. Even it allows domains to relax and supercoil independently. What does that mean? Think only this loop want to be relaxed. So as it is separately attached to this core, it can relax if it want, but leaving others still coiled. So it can relax independently by introducing single-stranded nicks, we call that, okay? And also it can coil independently. And this that ability to relax and supercoil independently is very important in transcription of specific genes. You will learn about transcription in protein synthesis, okay? But just remember, as that means that you can just only relax the part only you want, because you can introduce independent relaxations. So that's important. Also, you might have seen in the bacterial cells, there are circular DNA molecules called plasmids too. So they are also coiled and supercoiled. So that's for DNA packaging in prokaryotes. Now let's see DNA packaging in eukaryotes. It's actually a little bit complex than prokaryotes. It should be, right? Okay. We call the DNA plus histone proteins as chromatin in eukaryotes. Okay. Histone is a specific type of protein present in eukaryotes. So this chromatin can be further classified as euchromatin and heterochromatin. In euchromatin, they are lightly packed and rich in genes. So they might be active in transcription. Okay, simply transcription is making RNA from DNA. You will learn that in protein synthesis. For now, just remember that transcription is making RNA from DNA. It's necessary for protein synthesis. And when you go for heterochromatin, they are tightly packed and mostly inactive, okay? But they contribute in gene regulation, epigenetic inheritance, you learn that in genetics lesson, and also protection from chromosomal integrity. Now, we will learn the steps of, levels of DNA packaging in eukaryotes. It actually has 4 steps. So in the first step, you will have to learn something new called the nucleosome. Actually, nucleosome, my drawings are not very nice, but I tried my best to represent that. Here, I represent one histone protein from this circle. So, this has 8 histone proteins. Okay, one nucleosome has 8 histone proteins, and also this double helical DNA wound around that. So though I've just drawn it from single line, this is double helical DNA. Okay, so nucleosome is the sum of both of this. And to connect two nucleosomes, there's another double helical DNA part. We name that linker DNA. So this structure will form along this, okay? Nucleosome, linker DNA, nucleosome, linker DNA, nucleosome, linker DNA. In your resource book, you will have more clear images. Okay, so that's the first level, and the approximate height of a nucleosome will be 10 nanometers. Okay, in the second level, these the arrangement in first step is further coiled in spiral manner. See, it's like in a spiral manner to form the second level. Okay, so in second level, the height will be like 30 nanometers. It's just the first level is arranged in spiral manner and more compacted. And in the third level, we meet someone new. It's the protein scaffold. Here, this one. Now we have the this strand in second level. Already we have that second level thing, and it again form loops on the protein scaffold. Okay, this structured ones again form loops. So I represented in only the first loop. So that this type of a loop is called a looped domain. Okay, so that's the third level, and it will be around 300 nanometers. Then in the fourth level, what happens? The structure of third level further coils. See, even with the protein scaffold, it folds and folds and folds. So here, this thing is here. These are loop domains. I have not shown the nucleosomes because it's difficult to show it here. So it's again coiled, fold and more compacted. So this will be around 700 nanometers. And you know, chromosomes exist as 2 sister chromatids. So if one is 700, both will be 1400 nanometers approximately. So that is the DNA packaging in eukaryotes. So that's all. You will have to remember these. You might get to write short notes, even essays from this. Okay. That's all for this video. Let's meet with DNA replication in the next video. And if you found it pretty helpful, please give me a thumbs up, share, and don't forget to subscribe. Thank you for watching.